This video covers the multiple genes that form the immunoglobulins. Both the heavy chains, alpha, gamma, and delta, and the light chains, kappa and lambda, have a family of genes that are required to match the immunoglobulin variable region to an antigen for the immune system to work. Most of the genetic work on this has been done on mice, as their immune system is similar to ours. Each heavy chain and each light chain have a variable region at the outer end of the protein. The variable regions allow the immunoglobulin to attach to the antigen. This variable region is called the V region. The constant region is called the C region. These are linked together by a joining region, or the J region. The heavy chain also has a diversity region, or D region. All of these regions add to the diversity of the antibody. How can we generate antibodies for around 100 million pathogens without having 100 million genes? To do this with different gene sequences would make the human genome too large. The method our bodies use to generate new antibodies is through modular design. You make a new antibody by selecting from varying bits and pieces. In the human genome for the light chains, there are around 75 different V segments and 10 different J segments. To produce a heavy chain, you need a V, D, J, and C molecule. An immature B cell contains all the DNA modules, but when it links with an antigen, the mature B cell only contains one gene segment for each piece. For the light chains, kappa chains contain modules for the V, J, and C segments, while the lambda chains have J and C segments. The genome is a bit different for the heavy chain. There are about 45 different V segments and 6 different J segments. The heavy chains also have a D segment, of which there are 23 different genomes. So let's do the math. For a kappa light chain, there are 41 V segments and 5 J segments. This makes for 41 times 5, or 205 different combinations. For the lambda light chain, there are 33 V segments and 5 J segments, or 165 combinations. The heavy chains have 45 V regions, 23 D regions, and 6 J regions. Multiplied together, this allows for 6,210 combinations. Each antibody is made from one heavy chain and one light chain. So the full combination is 6,210 times 370, or over 2.3 times 10 to the 6th combinations. This is over 2 million possible combinations for an antibody to pick and choose from in order to make an antibody that matches the antigen. This is not quite enough to match the number of possible invading pathogens. So to add more diversity, when the gene segments are joined together, additional DNA bases can be added or subtracted to the final joint segment. This is known as junctional diversity, the changing of the antibody at the junction of the modular pieces. Now, the human immune system can make hundreds of millions of different variable regions to match whatever the antigen for the pathogen looks like. With over 3 billion B cells circulating in the human body, there are more than enough cells to produce the correct immunoglobulin. We only need about 30 B cells producing the same immunoglobulin to mount a good memory response. This concludes the video on Multigene Organization.